Thanks to the organizers and Illumina for giving me a chance to speak today. I am going to talk about how to train your Dragon for pathology informatics. Here's my obligatory slide to basically tell you all this is my opinion, not Illumina's, because this is a sponsored talk. So just a bit of background. I am now working as the laboratory director for XING Cancer Care, which is an accredited human pathology lab in Brisbane, Australia. We just got our accreditation in March this year, actually, right in the middle of the pandemic. Thank you, coronavirus, for killing all of our business. And I joined XING Technologies, which is our parent company, in mid-2019, mainly to work in the other part of our business, which is more about medical devices for point-of-care diagnostics. That is sort of part of the deal for me to come on board. I was asked to help push along our accreditation bid for our pathology lab to deliver an NGS-based capture panel. Not because I had any experience in pathology, but I had experience, I guess, in cancer genomics from my academic work in the last 10 years or so, and enough bioinformatics expertise, I guess, to kind of help push the process along. So I joined, you can see our company building here, our lovely medical scientist, Britt, working in the corner. The lab really started down this path in 2018, and it was largely led initially by academic scientists who didn't really get people experienced in pathology on board early enough in the process. So it was a bit of a bumpy road. Things came good though, but the timeline for us to get accredited was completely insane. So I said I joined in mid-2019. We were hoping to get our first audit in September. So I didn't really have a whole lot of time to get things going. And yeah, it was a bit of a challenge. So I'll just briefly introduce our tests. So as I said, it's a human pathology lab. We started with the XGP 1000 assay at the bottom. That's our just over 1,000 gene panel of genes that are known to be involved in cancer. In-house custom design, similar to the TSO, but obviously twice as many genes. And since then, we've actually developed a few more, another panel, which is an actionable cancer panel that just includes 72 of those 1,000. So it's very relevant and tight for cancer or cancer risk genes, and obviously much cheaper. And we also have a couple amplicon panels from another company that we're working with. So those were not designed in-house, but we are about to offer them as accredited panels. And just to, I guess, echo what some of the previous speakers were saying, we're starting to see, I think, a lot more people doing a paired tumor normal comparison, because it does really increase the resolution of variant calling. And we definitely do that for our tests because we do see a huge benefit. So I'm really happy to see that that's becoming a lot more common. So today I'm just going to be talking about these because this is what we started with, and I'll explain how Dragen has helped support our a bid for accreditation for these panels and how it works into our workflow. So just briefly, I hope everyone who's in the audience is kind of on board with this, so I don't have to talk too much about the background. We start with a solid tumor FFPE sample, usually from a slide, and macrodissect the region of that slide that contains concentrated tumor sections and extract DNA. And then we also get a blood sample from in an EDTA or a PAX gene tube, so we can get genomic DNA for the normal sample that we can compare with. Run it through our DNA capture panel, do the library prep, run it on a NextSeq 550. And in our case, long story, because we've done some quirky things with our panel, we can't actually demultiplex on the sequencer, unlike most people. So we actually send the BCL files to a Google Cloud virtual machine instance where we do some custom demultiplexing into FastQs. And then from there, we upload to BaseSpace. And I'll talk more about BaseSpace in a minute. If you are not weird like we are, you can make FASTQs right on a sequencer and go directly into BaseSpace. So you can skip the painful step of going to the cloud first. But luckily, because we do start in the cloud, the communication between our cloud instance and BaseSpace is super fast, ridiculously fast. So that is really nice for us to get things going. And then in BaseSpace, which is a cloud-based platform that hosts a number of apps that do various things, we can use the Dragen apps. Those are the only ones that I'm using for our pipeline. And there are 3, enrichment, somatic, and germline, which I'll explain in a minute. And those give us some useful information like coverage statistics about our 1,000 gene panel, somatic and germline variant calls, and of course, the aligned BAM files. Something interesting to note, we align against HG38, not my choice, I inherited this. It's made things a little bit difficult because, as you saw with the TSO 500 and probably a lot of other panels, they're still using HG19. And I think in the clinical world, this is pretty much still the standard. So there hasn't been a lot of support for 38 that I've seen. So thank goodness for me. Dragen supports that. I'm very grateful. And then we do a little bit of magic at the end and finally come up with our clinical report. So I don't know if many of you are around in, I think 2012, 2013, I think Illumina introduced BaseSpace around then. And sorry, Illumina, but when it first came out, I really didn't see the point. It, as a bioinformatician, I guess, it seemed quite useless to me. And I just sort of wrote it off and never looked at it again until I was given this ridiculous timeline to try to get our panel validated. And we were struggling to try to get GATK and the usual things running smoothly with good procedures and good documentation. And one of my colleagues just said one day, why don't we just use BaseSpace? And I really wanted to laugh in his face, but when he showed me how well it worked, I really couldn't argue because it completely saved our timeline and made this accreditation possible. So just to give you an example of our clinical reports before I go into how Dragen has been helping us, this is mainly, I think, for the people in the audience, if there are any who haven't been working in the clinical space. This is what our report would look like. So it's a fake patient. Don't worry about confidentiality here. I made up all the names and not all of the information here is real. But you can see here as a summary of the results, we have reported 4 alterations. So there's a somatic BRCA2, which we've said is clinically actionable, a couple somatic alterations, which are clinically significant, and a germline mutation alteration that we think is significant as well. We do report tumor mutation burden, so this one is fairly low. And then we report some clinical trials that this patient might be eligible for based on their molecular results. There aren't any approved therapies for this person, but we would have indicated them if we had any. Non-clinical people in the audience, you might wonder, why are we only reporting 4 alterations? Because based on a 1,000 gene panel, you would expect us to find a lot more. This is part of the magic. We're really only reporting the ones that are actually useful in a clinical sense that will either determine patient risk for cancer or hereditary risk that they might want to tell their family about, or alterations that can actually be treated with a targeted therapy. So that's part of the magic to filter out those things and to get something that's very pithy and doesn't waste too much of the oncologist's time in an interpretation. So how does Dragen help? As I mentioned, there are 3 apps that are hosted on BaseSpace. So the first one that I want to talk about is enrichment, and this is the one that delivers the coverage statistics. So it does a lot of analysis. You can see in the square on the right, there are many things that it can do: germline, small variant calling, somatic calling, copy number variant calling, structural variants, etc. And it delivers some sample and aggregate reports as well as some enrichment metrics. But really the only thing that I'm using in this case is the germline small variant calling, which I use for haplotype comparisons to make sure my tumor and normal samples are matched properly. And the metrics, I guess because I'm a command line junkie, I I like to do everything from text files rather from PDF reports, and it gives me a little bit more control. So this is a plot on the left where I've taken Dragen text file information, so just coverage statistics, generated my own plot. This is actually not mine, it's my bioinformatician who's done it for me, to show you a histogram of the single base read depths across the most important part of our panel, those 72 genes, not all 1,000, which is, I think, a nice little histogram. So that's a per level or per sample coverage. Here's a summary of the tumor sample, so you can get a more granular idea across that panel of the mean depth, median read depth, which genes, which are very clinically relevant for targeted therapies, are covered really well. We know we have a little bit of a BRAF problem, but it's not a part of the gene that is super important, so we kind of accept it at the moment. But this would really tell you if you are missing anything really worrying that the oncologist might want to know. If there's a alteration that you might have missed in one of these genes that could have a targeted therapy, you would either want to retest or let the oncologist know that you don't have any insight into that. Enrichment also lets you do per gene coverage, which is nice. So just a brief aside, anyone who's been through what Andrew was describing earlier, validating a gene panel for clinical use, they have to go through an accreditation process and an audit by NADA. And everybody I know dreads these audits because I think they are quite stressful, but I really enjoyed ours. I learned so much. We had amazing assessors. I'm really grateful actually for the feedback that we got. So one of our assessors, the bioinformatics assessor, recommended doing a hotspot analysis. So you can see here, this is the BRCA gene, BRCA1, and in blue, these are all the known hotspot mutations or hotspots where an alteration might be clinically significant. And so you can see here, because we've done this per base coverage, we see that all of the hotspots are covered really well, so we can be very confident that we're not missing anything that's really important. This is not something that would have actually been easy to do without Dragen enrichments per base coverage, because I'd have to use a separate tool, start up a new pipeline, and I could have done it. I looked into it, but it kind of broke me at the time. So I'm grateful that Illumina did it for me. And then we can also look at per target coverage, again, with this single base resolution, and not to air too much dirty laundry, but this is one of our regions that doesn't really work well. It's a bad set of probes. So you can see it tailing off quite badly down to 0 coverage at the end. But it's really important for us to be able to see this and know that we don't really have any insight there. Yeah, so that's Dragen enrichment. We do a little bit more reporting. This is all part of our QC to decide if our sample was run well or not. And then Dragen somatic is maybe the most important part. This is where we get somatic alteration calling. And as I said earlier, we do tend to do tumor normal comparisons because that gives us better resolution into true somatic variants. You can also run a tumor-only pipeline, which we did do during our validation when we didn't have paired samples. But obviously, you can make it work for whatever pipeline that you're running in your own lab if you choose to do that. And this basically gives you your somatic VCF and your BAM at the end. And then Dragen Germline, which is a third app that we use and run only on the normal sample, just gives you germline alteration calling. So there's a VCF and a BAM at the end of that. And just to be, I guess, very honest, and if anyone is considering looking at this, I did kind of run into a problem when we participated in a quality assurance program for DNA NGS testing. I don't have a strong background in genetics, so I never look at the genotype field in a VCF. So if anyone is out there is used to looking at VCFs, here are a couple lines. Out of one of them, there are 2 variants that I've shown at the top, chromosome 1, chromosome 2, different positions. These are 2 variants that were, 2 alterations that were called as germline, and Dragen germline has reported the genotype shown in red as either heterozygous or homozygous, which is great. However, Dragen Somatic, which is the alteration on the bottom from chromosome 3, oops, sorry, the bar is obscuring my screen. I can't quite see what's underneath it right now. But the Dragen Somatic genotyping is always 0/1. Coming from research, I never look at the genotype. I always just look at the variant allele frequency. And because I've always ever worked with tumor normals, I don't really need to worry about whether it's a germline or a somatic. So I got caught out on this because I ignored that field. So just something to be aware of. I guess Dragen Somatic does some strange things, I guess. I would call them strange anyway for the genotyping in the somatic. Anyway, otherwise, it's all good stuff. And whoops. There we go. So just to show you a little bit about what BaseSpace looks like, most people, I guess, who are not command line junkies like me would like to use the BaseSpace Sequence Hub web interface, which is what this is. This is the Dragen enrichment app that I've pulled up just to give you an example. Lots of mouse clicking to enter your stuff. So after you upload your FASTQs, you can select those as a biosample, give them a project to assign the results to. And eventually there's a lot more stuff at the bottom of here. You submit the app and it runs in the background, and then you get an email when it's done, and then you can go look at the results. It works really well, and this is what I did during our validation and for the audit. But I hate clicking, so fortunately, BaseSpace offers a command line interface, which has been amazing. And again, I'm not expecting you guys to look at all of this, especially if you're not bioinformaticians like I was. But I just wanted to kind of show you how simple this command line interface actually made the whole pipeline. So this literally is every command that we run for a batch of 5 to 6 patients for our 1,000 gene panel. So basically it takes 5 to 10 minutes to create a project, upload the FASTQ files. This is after they've been demultiplexed. 8 to 10 minutes to run Dragen Somatic, about 15 minutes to run Dragen Germline, but they can run in parallel. About 30 minutes to run Dragen Enrichment. And again, this is all just one command per app. And then once everything is done, you can download the whole project. And because it comes from the cloud, if you've got a good internet connection, the whole thing comes down in about 20 minutes. So this takes about an hour for a full flow cell, which would be 5 to 6 patients for us, which I think is pretty fast. I was pleased with that anyway, considering the time that it takes to actually go through the capture and doing all the sequencing. The amount of time to do this analysis is basically negligible. So the thing about this, I guess it's a bunch of command lines that, or command line commands that a person would have to run. We've wrapped this all into a shell script. So you can just basically push a button and it runs everything and downloads all the files into the right place. And one thing I haven't actually looked at is in depth the documentation for the command line interface, because there's a little thing down here which is workflow. I should probably talk to Illumina about this, but I just haven't had time. I'm kind of thinking that maybe if I use this workflow, it will do something nice for me. So I'll get my bioinformatician to look at that at some point. That sounds promising. But otherwise, it's been pretty easy to take all of these and just wrap it into, I think it took him a couple days just to write it all up and test it and make sure it all worked. So pretty easy to use. And if you are afraid of command line, you can always do this. It's pretty easy as well. It just takes more clicking. And then again, if you're working in clinical labs, you'll be aware that obviously patient confidentiality is of critical importance. The ISO standard for 15189 specifically states that patient samples have to be guarded from unauthorized access. And the NPAC Tier 3 document, again, that's specific to Australia, says that you cannot disclose any confidential information for a patient. So BaseSpace isn't actually meant for this kind of thing, but they do have some security practices in place. And when we use this, we obviously de-identify everything. So no patient information goes up to BaseSpace, just to make sure that everybody is protected. And in fact, I try to de-identify these samples as much as possible, even in our own hands, because just safer that way. Oh, and then the most important thing, I could not believe how, sorry, I'm really sounding like I'm trying to sell this. I'm not. I just could not believe how cheap BaseSpace was. So when my colleague said, oh, let's use BaseSpace, I instantly thought, this is gonna cost 20 grand a year. It's gonna be horrible. I don't think I can afford it. But when we actually looked into it, you can see in the red box up here, 5 iCredits per node hour. I actually calculated the cost just of running Dragen Somatic at the time, and I think it came out to $1.32 per sample or per flow cell, I can't remember. It was ridiculous. So that was pretty much the nail in the coffin for, not the nail in the coffin, the deciding factor for me. Super cheap, super fast, and I didn't have to do almost any extra development work. So that was how we were able to make our ridiculous timeline. Our audit actually got pushed to October, but I was fully ready and pretty impressed that I was able to do this so quickly and still quite cheaply. Just to give you some, I guess, not really benchmarking, but comparison results to show you that it's working nicely. This was the version of Dragen that Dragen Somatic that I started with, which was 3.3.7. We've moved on a little bit since then, but this was part of our validation report. It's not exactly benchmarking, but these are clinical samples where we know what the clinically significant alteration was. And you can see these are all colorectal cancer samples, varying different tumor percentages. And you can see the reference allele frequency and then our allele frequency. And basically we got them all right. So we knew that Dragen Somatic was working really well, even at sort of lowish tumor cell percentages. Of course, we used some reference samples, which I think Andrew referred to as well earlier. This was the Horizon OncoSpan, which is, I think, a pretty famous one. This is, I think, a cell line that's either been artificially made to look like this, but you just purchase this, and all of the allele frequencies have been tested by either sequencing or droplet digital PCR, so they're pretty well characterized and accurate. And you can see the list of alterations down the left, and then in our replicates, we've done three replicates here, and you can see that we've got 100% concordance between everything, and the allele frequencies are actually quite correlated as well. So again, that's telling us Dragen is working pretty well. And then here's an example of an EMQN scheme. So this, again, for people who are not working in the clinical space, this is a quality assurance program. So we were sent a number of samples that were blinded. So we just had to test them, see what we came up with, report the variants or alterations as we found them. And again, we did really well. So that speaks to our panel, I guess, but also to the bioinformatics that we use to call these variants. Yep, sorry.
Yep, you go for it.
Oh, yeah, that was the end. I just wanted to acknowledge my team. We're a small crew because we're just a startup company. And yeah, you can see them listed. That's all. I'm happy to take questions.